Hello, hello. जैसे लेक्चर चालू होगा ना वैसे लोग इस सीट से
Good morning, everyone. I would request our esteemed dignitaries and guests to kindly take seat on the dais. Dr. Roshan Shankaria, IAS, Professor Uttam Raj Bhandari, Professor Arun Grover, Dr. Dajinder Kaur Arora, Dr. T.R. Sharma, and Professor Hirendra Ghosh. We would like to give floral welcome to our dignitaries. I would request Aarti to present bouquet to Professor Uttam Raj Bhandari. To Dr. Roshan Shankaria. To Professor Arun Grover, to Dr. Jatinder Arora, to Dr. T.R. Sharma, to Professor Hirendra Ghosh. I request you all to kindly switch off your mobile phones or put it on silent mode. Distinguished dignitaries, delegates, scientists, teachers, and students, it gives us a great pleasure to extend you all a warm welcome on behalf of Punjab State Council for Science and Technology, Institute of Nanoscience and Technology, and National Agri-Food Biotechnology Institute for this third INST PSCST Hargobind Khurana lecture. INST, in association with Punjab State Council for Science and Technology, instituted a lecture in the field of bioscience to commemorate Nobel laureate Professor Hargobind Khurana, a world-renowned biochemist. The objective of this series is to provide a deeper insight into the realm of science to students and teachers in and around Chandigarh region. We are fortunate to have Professor Uttam Raj Bhandari, Lester Wolfe, Professor of Molecular Biology, MIT, who has been kind enough for delivering this third INST PSCST Hargobind Khurana lecture. Professor Bhandari is an eminent personality and is a very close associate of Professor Hargobind Khurana. As we all know, Professor Hargobind Khurana has done revolutionary work on the interpretation of genetic code and its function in protein synthesis. His work has opened up new discipline of research in biology known as genetic engineering. I am delighted to share that we have 100+ students, especially from schools of Punjab, who are present here to hear a lecture of Dr. Raj Bhandari. Now, I would like to request Dr. T.R. Sharma, Executive Director NABI, to kindly deliver a welcome talk.
Very good morning to each one of you. Distinguished Speaker of the Day, Dr. Uttam Raj Bhandari, Lester Professor of Molecular Biology, MIT, USA. Chief guest of the function, Dr. Roshan Sankaria, Principal Secretary, Science and Technology and Environment, Government of Punjab. Professor Arun Kumar Grover, Vice Chancellor, Punjab University, Chandigarh. My colleague, Dr. Jatinder Kaur Arora, Executive Director, Punjab State Council for Science and Technology. Dr. H.N. Ghosh, distinguished guest, Khurana Associates, directors of different institutes situated at Chandigarh, students and teachers from different schools, universities, NABI, CIAB, INST, ladies and gentlemen, press and media. It's indeed a matter of great privilege for me to welcome you all for this very important series of lecture in the memory of Professor Hargobind Khurana, the Nobel Laureate. First of all, I would like to welcome Dr. Uttam Raj Bhandari on behalf of NABI as well as my other sister institute, INST and Punjab State Council for Science and Technology, for agreeing to deliver this lecture. Most welcome, sir. From my personal behalf as well as on behalf of all of us, I would like to welcome you to this beautiful city of India, Chandigarh. I would also like to welcome Dr. Roshan Sankaria ji for agreeing to be the chief guest of this important series of lecture which Punjab State Science and Technology Council has started in collaboration with INST as well as NABI. My special thanks are and welcome to Professor Arun Kumar Grover ji, who is not only Vice Chancellor of Punjab University, but also he is like teacher for all of us, those who are new to this particular city, all these directors. He is the senior most faculty and director or vice chancellor of among all the 21 institutions situated in this region. And the most important thing which I would like to welcome here, Professor Grover is guiding us from the top in the form of a consortium of all the institutions which he has developed with a novel idea called CRIC. Probably he will say in his own talk in details about that. And that's very unique idea which he has implemented along with all these institutions. My special welcome are to the students and teachers from different schools, colleges from Punjab. This lecture is basically for you and for all of us. If you remember and if you know a little bit about Professor Khurana, but I would say that the saying is simple living and high thinking that goes with our Nobel laureate, Dr. Khurana. We were having 3 days conference, and you can see this board on the side on gene, genome and molecular biology, membrane biology, by more than 24 Khurana associates who came from different parts of the world during these 3 days. And what I came to know from all of them, Professor Khurana was really a doer, a very hardworking fellow who works 24/7, coming from a very small village of Punjab. That's very important to note. From a very small village of Punjab, from a very humble background, he rose to this particular position by receiving Hargobind, this Nobel laureate, because of his own hard work, sincerity, honesty, and integrity. That's what I would like to say. Only one word to my students from different schools and colleges, that whatever you do, you should be happy to doing that, and whatever you wish to do, you should follow that path with more honesty, integrity, and sincerity. No one can stop you to achieve your goal. With these words, I would like to again welcome you all for this very important session, and we would be having a very nice lecture from Professor Uttam Raj Bhandari. Thank you very much.
Thank you very much, sir, for your kind and inspirational words. Now, I request Professor Hirendra Ghosh, Scientist G, INST, to kindly introduce the speaker.
Good morning, everybody. It's like reintroducing Professor Raj Bhandari again and again. I'm so privileged, sir, to introduce you in this august audience. Well, Dr. Uttam Raj Bhandari was born in Nepal. After his early education from Patan School in Nepal, he received his MSc in chemistry from Presidency College, University of Calcutta. He was gold medalist. And PhD from University of Durham, England, with Professor Sir James A. Baddeley on synthesis of compounds related to coenzyme A, including polymer from bacterial cell wall. In 1962, he moved to University of Wisconsin-Madison, USA for postdoctoral research with Professor Hargobind Khurana. In 1969, he joined as a faculty of the Department of Biology at Massachusetts Institute of Technology, which is MIT at Cambridge, where he currently holds the Lester Wolf Chair of Molecular Biology. He and his research team, including graduate students, postdoc fellows, and visiting faculties, have working extensively on nucleic acid and nucleic acid protein interactions using biochemical and genetic approaches. He also worked at the National Cancer Research Institute in Tokyo, Japan. Dr. Raj Bhandari has published more than 230 papers, research papers, in top scientific journals, many of them like PNAS, Science and Nature, which is pretty much elusive for Indian scientists. For his outstanding contributions in molecular biology, he has been elected to the American Academy of Arts and Science, and he has received many other awards. Dr. Raj Bhandari has received on the editorial board, served as the editorial board of many prestigious national and international journals like Archives of Biochemistry, Biophysics, Nucleic Acid Research, Analytical Biochemistry, Journal of Biological Chemistry, and Gene Expression. With this brief introduction, we are welcoming Professor Uttam Raj Bhandari.
I request esteemed dignitaries and guests on the dais to kindly take seat with the audience.
Thank you. Great. Wonderful. Thank you. Yes, thank you. Dr. Arora, Dr. Sharma, friends and colleagues, and all the students who are here. It's a great honor for me to be giving the Gobind Khurana lecture today, named after a scientist who was a pioneer and a visionary and one of the greatest scientists of the 20th century. I thank Drs. Arora, Sharma, and Ganguly for this invitation. And I thank all of you for coming to this talk. It's a challenging and almost impossible task
to be following the likes of Venkataraman Ramakrishnan and Gurdev Singh Ghosh. And I'm truly humbled by this opportunity to be presenting this lecture to you. Dr. Khurana's scientific journey began at Punjab University in Lahore, where he obtained the degrees of BSc and MSc. From there, he went to Liverpool University in England to obtain a PhD degree in chemistry. And like all other academic scientists of that era, followed this with postdoctoral work, first at the ETH, the University of Science and Technology at Zurich, and then at Cambridge University in England. He then started his own independent research at the British Columbia Research Council in Vancouver, became well known for his work there, and after 8 years, moved to Madison, Wisconsin, where he established the genetic code, for which he was awarded the Nobel Prize. While there, he also synthesized a gene for the first time. Another scientific accomplishment in genetics. After 10 years at the University of Wisconsin, he moved to MIT Cambridge, where he spent the rest of his scientific career. While at MIT, he demonstrated again for the first time that a synthetic gene, a different gene that he had made, could be introduced into cell and could be shown to be fully functional in the cells. And this was literally the beginning of genetic engineering. That's what really started working on genetic engineering, and that's what we really do use all the time now. He then started working on a completely new area of research on membranes and membrane proteins, publishing more than 250 papers over a period of 30 years, and again doing trailblazing work and making fundamental contributions. Now, at most lectures of this type, it's customary for the speaker to, after paying homage to the person after whom the lectureship is named, to go to a description of his or her own lectures or work. However, I would like to do something different. I would like to spend more time talking about Dr. Khurana himself, his work ethics, his scientific accomplishments and all that, because I've had the good fortune of having really known him for a period of 49 plus years. A very close association. I had the privilege of having a very, very close association with Dr. Khurana. And I believe that based on my conversations with him, based on the papers of his that I've read, many, many papers of his that I've read, I know him, something about him, Khurana the man, his scientific goals, his approach to science, his work ethic, and how he overcame many, many barriers and problems that he faced along the way. I would like to share that information with stories and anecdotes about the remarkable scientific journey of this man, so the audience here, particularly the students here, can understand how a person with such a starting from a humble beginning became such a great scientist and one who was admired by so many. Now, to give a little background, Dr. Sharma has already mentioned this. Gobind, I will now call him Gobind. I hope you're not upset by this, because I don't want you to think that I'm being too familiar. But Gobind was the name we called him all the time. The first day I met him, he said, "Call me Gobind," and that's what we did. So Gobind grew up in Raipur, a very small village in the Multan district of Punjab, consisting of about 100 inhabitants. And his family was the only family that was literate in that whole village. There was no building for an elementary school. Elementary school was basically under a tree. The ground under a tree was the elementary school. His father was a patwari and a low-level agricultural taxation clerk, and the family lived an extremely humble life. So how a person who grew up under such humble circumstances, could rise to become an icon of molecular biology and one of the greatest scientists of the 20th century is a fascinating story in science. If we can understand how he overcame the barriers that he faced and yet became so successful, we can learn from it, perhaps, be inspired by it, and emulate him and be successful in our own adventures and pursuits. Now, let me first tell you how I got to know him so well personally. My association with Gobind started in 1962 when I received a letter from him offering me a postdoctoral fellowship. I was very happy to accept that, so I went there and met with him. And then I joined the lab in October of 1962. And after 7 years together, working together in Madison, we both left Madison to go to MIT in 1970. I left in 1969, he came in 1970. And our close association has really lasted until his passing away in 2011. So I've known him for 49 plus years. At MIT, our research laboratories were next to one another, they were adjoining. We shared the research facilities and equipments, and our two laboratories essentially function as one, even though we worked on different scientific projects. We also had joint lab meetings, so I got to know what people in his lab were doing, and he got to know what people in my lab were doing. We also had joint lab outings and social gatherings, and this led to a very close association between his group and my group, and of course between him and me. So to make this point, I'll now show you some lab photos of Gobind's and mine taken over the years. So I'm going to show you a whole bunch of photos now. We'll go through one by one. First, this is now a world view of Gobind's scientific journey here on a world map, starting in Punjab here, Lahore, Punjab University, went to Liverpool, got his PhD, went to Etihad, a short postdoctoral stint, we'll have more to say about that, Cambridge, UK for 3 years, went to Vancouver, 8 years, Madison, 10 years, where he got a Nobel Prize, came to Cambridge, MIT, where he's literally stayed all the rest of his life. All right? So, this now is a photo taken in Zurich in 1948 when he was a newly arrived postdoctoral fellow, and you can see him at the corner here. This is Gobind Khurana. And his professor, I believe, is this one here, Professor Prelog. So, you know, that's what's the first, his first photograph taken at the Etihad. And then this is now, he has gone to Vancouver, and now he has his own research group, and this is now his research group, where he is doing whatever he wants to do, and this is him now. And this is the group that he accumulated quite quickly and became very, very successful in Vancouver. And this is a person, Mike Smith, one of his postdoctoral fellows, who went on to win the Nobel Prize himself later on. He stayed on in Vancouver and went on to win the Nobel Prize. Yeah. Now, and now we go to Wisconsin, Madison, Wisconsin, an Institute for Enzyme Research here, enzyme research. And now this is his research group. All of them basically with lab coats, except for one. And let me see where Gobind is. Gobind is here. Yeah, okay. This was in 1960s. It was taken in 1964, I think. And then the next one in 1965, basically the same thing. Some new people, but basically same. And then we have 1970s. Now we start at MIT. So we have, this is our joint group now in the 1970s. Again, Gobind is here. 1970s, 1980s. Okay, here is Gobind, kneeling. Yeah. He was a very humble man, you know, he would sit, kneel, whatever. He was with his group and he was happy, that's all it was. Now this in the 90s, group is getting a little smaller, but still, you know, we're all together. And this is him here. 2006. Now we are fewer, but still, Gobind here. And we also had joint PhD students. And here I show a PhD student, very happy, immediately after defending his PhD thesis. So this is a kind of after thesis party, if you like. And this is of course, this is Gobind and this is me. So we had, he was one of our joint PhD students. And we also, Gobind loved to be with the group, wherever he was. So we had lab outings either at his place or at my place, wherever. And this was a lab outing about 2 miles, 2 hours drive away from MIT, where they had, Gobind and his wife had a small cabin, but we went there almost every year around apple picking time. We went there and we would go pick apples after the party, lab party. And here he's playing volleyball. And so long as he's doing it together with the students and his lab group, he was happy. That's what he did. And then here we are hiking up in the mountains. Again, he loved to go for long walks and hiking and all that, and he's here. Yeah, with whoever in our 2 groups wanted to go hiking. And then that's later on, about 2000, something like that. And this is even later, about 2010, 2009. Oh no, it's here. Okay. So here he's relaxing at his summer place in a place called Rockport, about an hour away from MIT. Okay. Now, he also was incredibly kind and generous to me. He would write short notes. Whenever he published a paper, he would just write a note saying to Tom with, so and so. And he left many, many notes like that. I cherish that. But I show you a couple of them. And this is now a copy of his Nobel lecture in December 12, 1968. And he writes to Tom and Gobind. Right. And yet another one. And this is it shows how kind he was. He said, this is now after one of my colleagues, who also was a postdoc of Gobind's, he and I, in 1993, edited a book together. It was published under the auspices of American Society of Microbiology. We edited a book. The book was published, and there was a review of the book. And Gobind saw the review of the book before we did, and he left me this very nice note, very encouraging note. He said, Tom, you saw the wonderful review of your book in Nature Structural Biology with a question mark, Gobind. And it was left penned on my office door, which is right across the hall from his office. And he would do that. I either just pen it on my door or leave it with my secretary or whatever. Many, many such things. So I really cherish these things. So this is really what Gobind was as a man. Now, he, at Punjab, right? So the question is, how did he, as someone who really started with such humble beginnings, even get to a university? Yeah? Well, he got there through sheer determination and hard work and self-motivation. And this is something that Dr. Sharma has already told you what our students should be really doing. One of the most important lessons to learn from his success is be determined in what you want to do. Work hard, right? And be self-motivated. Someone doesn't have to tell you to work hard. You work hard on your own. So with that kind of thing, he eventually made it to Punjab University. And he even published 2 papers. Three, okay. I have well, I have all 3. You have also, I have only 2, unfortunately. Okay, published 3 papers. That's even better, right? Yeah. Okay. So this is a paper that Gurbakh Singh, I think, sent it to him in 1995, and he has made a note, say part 3 is the next paper. And then this is the part 3. And in the first one, actually, we don't even have his full name. It just says Har Gobind. Yeah.
They were both classmates.
Gurbakh Singh and yeah, that's right. Yeah, that's right. Yeah. Okay. And Mohan Singh was the boss, I think. Yeah, he was the PI, right? So that's yeah. Now, so I think we talked about the sheer determination and all that, and Gobind had many characteristics that I thought were really important. They were the guiding light. These were his scientific mottos, if you like. Yeah. The important thing is, be determined and self-motivated. And the next thing is be bold and aim high. You know, you really try to go for the sky, if you like. You know, to go as high as possible. Nothing is impossible is basically what he would say. And I think he often told me this: if you want to get far, you have to travel alone. And so he was not frightened to be way on the wing, right? Do whatever he wanted to do. And in fact, he tried to do things that at the beginning looked impossible to do. And yet, he tried it, and he succeeded every time he tried it. Be disciplined and work hard. Incredibly disciplined, very hardworking. Always do your best. Strive for excellence. And this was another motto. He was, everything that he did, science, whatever, playing volleyball or doing other things with it. Yeah. If he was challenged, he would accept the challenge and he would do it. And in fact, one of the things that one of his colleagues from Vancouver said when he got the Nobel Prize was, he showed us what excellence in science was, and we learned to recognize it. And that was really that you learned from him. He did not tell you what to do. He just provided an example of what should be done. And stay focused and don't get distracted. Do not get distracted. We will have an example of that, his focus. Plan carefully, prepare yourself well. And whenever you start on a new project or something like that, you would plan meticulously. He was meticulous about planning. You made sure everything is okay. If you needed help from outside, he would start collaborating with people outside to do that. But whatever he did, he succeeded because he planned things very carefully. Be thorough and follow every lead. In your experiments and things like that, planning for your to do experiments, execution of your experiments, be thorough. No shortcuts. Do everything necessary to prove a point. And another very important thing, persevere, do not give up. He never gave up. Yeah. And if things don't work, you try hard. You try it again. And you try different ways of achieving a goal. Eventually, you will succeed. So this is his motto. And I think many of us, we all try to live by it. And okay, so now, along with the... where do I stand? Yeah. Now, we go from here to his stay in Liverpool. So he went from Punjab to Liverpool to do his PhD. The work was going well, but he realized that there was something wrong. Basically, when you do science, you have to learn to communicate with your peers, with anybody you're talking to about what you're doing in the lab, why you're doing it, yeah? What are the results you got, and how are you interpreting it? And so, in other words, to explain the science that you're doing to people around you. And he realized that there was some problem, that he was not really very successful at doing that. And the reason for it is he realized that in terms of speaking style, mannerisms and all that, we go from this part of the world to Europe or to US, and we have a habit of speaking very, very fast. And people in the West find it very difficult to follow, speak very fast, speaking fast. The other thing are the mannerisms some of the speaking manners that we use. And the third thing, of course, is our accent. Our accents are not quite the same as that of others. And so in other words, you can't communicate effectively unless you can really speak or talk about the person who is listening to you. And he realized that. And he told me that he overcame that really in a very, very interesting way. He was absolutely determined he was going to overcome it because he found out that this was getting to be a kind of a barrier for him. Yeah. So what he did, he told me, was instead of going out with his buddies, his classmates, on Friday nights and Saturday nights, which is the tradition in England, Friday nights and Saturday nights, they all go out, to eat and play sports or whatever, or just generally enjoy it. He stayed home in his room that was cold in England, in his one-room apartment, nothing more than that, and he listened to the BBC Third program, radio. The BBC Radio Third program had a program where they basically talk about things, interesting things, and they speak it in such a way that if you try to emulate it, you learn how to speak correctly and slowly. So that's what he did, and he conquered it. He conquered the problem that he thought he had. And he was one of the most effective communicators, oral communicators. And then he also realized that his oral communication is good, but also you need to do a better job of the written communication. Because after all, you do science, you write the paper so that others can read it. And if you can't write it effectively, first you may not get it published, right, which is not good. And even if it's published, if it's not clear, people ignore it. Right? And so he found that he had to do something, and he read, so Bertrand Russell, a great English philosopher, was one of his heroes. So he read Bertrand Russell's books whenever he could. And he got something from the book that he got. So these are excerpts from How I Write by Sir Bertrand Russell. 1, never use a long word if a short word will do. 2, if you want to make a statement with a great many qualifications, put some of the qualifications in separate sentences. Don't put it all in one sentence and make it completely garbled. Right? And 3rd is, do not let the beginning of your sentence lead the reader to an expectation which is contradicted by the end. If the two are different, then you really are not making any sense by that sentence. Right? So he believed very, very much in that style of written communication. And I have to tell you that if you read his papers, you'll find that, number 1, the sentences are short. They're very precise, no fancy big words, very precise. They're very clear, and it's a joy to read. And so the barrier, in terms of written communication was also gone. And so that's the kind of thing that he would do to achieve a goal that he set for himself, because he found it was necessary to achieve his bigger goal of being successful in science and in life. Now, so from Liverpool, then he went to ETH in Zurich. And the reason for going to Zurich was really that when he was a chemist, and he had to read many, many papers in chemistry. And he found that many of the papers were in German. And so that became a barrier. So when he decided, when he finished in Liverpool, he decided that he really wanted to go to a country where they spoke German. And so either Germany or Austria or Switzerland were the 3 countries. And Germany, just after the war, was not possible. So he decided to go to Switzerland. But now, if you want to find a place to do postdoctoral research, what you normally do these days is you apply to the professor you want to work with a year in advance or something like that. And then you wait and hope that he or she will accept you. Yeah. But Govind did not do that. He just took the risk that he wanted to work with a certain professor in Zurich. And so he just took the train with his PhD thesis in hand and went to Switzerland. And this is what he says. He actually says this, Okay. He says, I arrived in Zurich one day in late summer of 1948. I left my luggage at the railway station, took my PhD thesis with me, and went looking for Professor Prelog. He had no idea then that an Indian student was about to approach him to plead for a little space in his laboratory. No one from Liverpool had sent out any letter of introduction or recommendation on my behalf. When I met Professor Prelog, I found him so accessible that I immediately lost all fear. I requested that he might look at my PhD thesis. This was an organic nitrogen compounds and alkaloids, and I would come back the next day to ask if he would accept me. Next day, Professor Prelog, with his typically enthusiastic and encouraging smile, said yes, and at my excitement added that he was glad to see people happy. Looking back now over the years, I believe that spending a year at the ETH with Professor Prelog is probably the wisest thing I ever did in my life. And that indeed was true. And he stayed there for only 11 months. And that's a very short time to stay in a lab. And the reason for 11 months is because Prelog was not able to support him financially. So he supported himself financially with money that was left over from saving when he was a student. And all he could afford was 11 months of living in Switzerland. And so he was willing to sacrifice everything that he had to, again, go to a lab where he could do some work and also learn the German language. Another extent of dedication. And then, so he published papers there in German. You can see that. And his co-authors are Prelog, who is the professor, Pralog was to go on to win the Nobel Prize after Khurana. Govind won the Nobel Prize, but he won the Nobel Prize later on. Khurana and G.W. Kenner, I'll mention that because that name really will come back later. So this is one paper, and this is second paper. So 2 papers in 1 year is very, very good work, right? So now, so he finished that. All right. So another thing that he did was, he saw that he would really learn German language and also communicate in German, give seminars in German and things like that. If he were to really go and translate papers written in German, and so that he could really feel comfortable about that. So he did that. He says that. In between doing experiments, you'd go to the library. The library happened to be close by. Go to the library and start translating papers in German. And then he realized that he came across a compound called dicyclohexyl carbodiamide in one of the papers. And he read it, and he found that very interesting. And he discovered a reagent that had been originally found in 1870. That was 80 years ago or something like that. But then he realized that this was a very interesting reagent. And he remembered that. And that reagent was to be the foundation for the work that won him the Nobel Prize. That was the foundation for his work on that led to the synthesis of a gene. That simple reagent that he discovered while he was translating papers in German, from German to English, because he wanted to learn the German language. So this was another extent of his determination to want to achieve things and how he would do it. Okay, so he finished at ETH. He was happy, but he could only stay there for 11 months. Now he had to find a job. So he comes to India. This was 1950, I think. 1949. This was soon after independence. His family had been uprooted from Lahore. India was in chaos. And so he couldn't find a job. But fortunately, while he was at ETH, he established a close friendship with George Kenner, who was his co-author of the paper. And George Kenner also had gone back from ETH to Cambridge University. And so the two of them communicated, and Kenner said, I'll talk to Todd and get you a position here. So he found a position in Todd's laboratory at Cambridge University. And he told me, actually, that the family had to somehow gather some dollars, some pounds, for him to get the boat from wherever he was to catch the boat from to England. And because the family was literally, this is really soon after partition, and it was very, very humble. They were living humbly themselves. Yeah. So anyway, but he got his way. He got to Cambridge, and worked there, and for 3 years was happy. He also tried this DCC reagent in one experiment, and the result was so interesting, so important, that Todd published a paper right away on it and was enormously impressed with him, because he was able to achieve in one experiment that the Todd lab had been trying to achieve for many, many years. And this is in a lab who went to win the Nobel Prize for chemistry. And this is really what he was able to achieve based on his discovery of this reagent called DCC. And now he describes now what happened here. After 3 years at Cambridge, he says, he was trying to find a job wherever he could. He didn't care where he went, but he wanted to have a job where he could do research of his own that he wanted to do. And fortunately, Gordon Strum, who is a Canadian, was trying to recruit people to Vancouver. I was interviewed by him. I didn't know exactly where Vancouver was, but I agreed to go there if Strum would offer me a job. Gordon Strum had obtained a small grant and wanted to see some fundamental research started there. Gordon Strum, who is a physicist by training, also told me quite candidly that he was looking for an organic chemist because he thought organic chemical research was the cheapest to carry out, requiring only test tubes. So with this not so auspicious offer of a job, and not knowing where Vancouver was, he still accepted the job because it allowed him, it was the first thing that he wanted to be independent to his own research. Yeah. And he took that job and went to Vancouver. And he did phenomenally well. He established such a reputation there in Vancouver that much happened. Yeah, and I'll just describe to you what he was able to achieve in Vancouver. He used the organic chemical synthesis to synthesize compounds that were very, very important biochemically, and yet could not be made in large amounts. There were no companies that made these large amounts, and so you had to establish methods that allow you to make these compounds. And so he learned to use the chemistry that he had learned, and also the DCC as a reagent, that DCC that he discovered as a reagent. He made something called ATP, which is a very important biochemical compound, coenzyme A, and we'll get back to this later, coenzyme A, a very important coenzyme, and people have been trying to make it for years, and not succeeded except him. And then these compounds. So these are the important compounds that he made, including even the first beginning of a nucleic acid that he was able to make. And so as he published those papers, biochemists elsewhere took notice. And they realized that here was a guy, here was a person who basically is a chemist, but really doing work on biochemical problems, biological problems. Right? And so they were interested now in coming to his lab. They would take summers. The people, well-known, established biochemists would take summers off, bring their families to Vancouver, and then they would work in his lab, learning from him what he could teach, and also teaching him, the biochemistry and biology in general. And so it was a very good way of communication between two research groups. And so people like, he had people like Arthur Kornberg and Paul Berg, who themselves were to go on and win the Nobel Prize, Paul Berg and Arthur Kornberg. And they came. They worked in his lab. They made things that were very important for them. They took it back to their lab. And Arthur Kornberg made something called deoxynucleoside triphosphates, took it back, because that's what he used to work on DNA replication. He's the god of DNA replication, if you like, Arthur Kornberg. And that's what he used. He synthesized those compounds in Gobin's lab, took it back so he could use it for his studies of DNA replication. So the result is that there was this good communication between the two research groups. They became lifelong friends. There was networking established, and it mutually beneficial to both groups, or all three groups, because Paul Berg has his own group. These were guys from Stanford University. So here I show a picture that I think I took of a meeting, one of the same kind of meeting that we had here the other day, yesterday and the day before. That was in Vancouver. This was a 1993 meeting in Vancouver. And here, oops, this is, no. Yeah, 1993 meeting in Vancouver, and this was some kind of gathering outside the auditorium. And they became lifelong friends, and so much so that this many years later, 2006, maybe 50 years after, the two of you see Gobin and Arthur Kornberg in Arthur's house. Yeah, they're just almost like children playing together. There was just so much, such friendship. Okay, now, so then he moved to Wisconsin, and this is where he did the work with genetic code. And the idea for him, again, the idea that he wanted to use is simple. He always wanted to do things his own way, his own approach, because he felt that this was his strength and he should exploit it. And his strength was in being able to chemically synthesize material, right? So he did that, and he used this is where DCC again came in. He used DCC to make very, very short DNA molecules, DC, TC, 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 very simple things. Same thing repeating over and over again, right? And AG, 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 AG. And if you mix the two together, what happens is that the two come together through what's called Watson-Crick pairing. You students know Watson-Crick story of Watson Crick, DNA structure, right? A pairs with T and G pairs with C. And so when you add these together, these two together, and you have T binds to A, C binds to G, T binds to S, so that's how the two come together. And then you can convert this very small molecule into a big molecule using an enzyme called DNA polymerase. And you can convert that long DNA to an RNA molecule using an enzyme called RNA polymerase. And RNA polymerase will have the same nucleotide sequence as the one of the strands of the DNA has, right? And then if you now, this is a very simple RNA, if you now use that to make proteins in an extract in the test tube, and you can analyze the protein that you have made, you have essentially established the genetic code. And he did that with many, many different kinds of polymers that he made to establish it. That was his approach to establishing the genetic code. Now, of course, here again, the friendship with Arthur Kornberg and others came extremely handy. Because for DNA polymerase, he didn't know how to make DNA polymerase, but Arthur did, Kornberg did, right? So he went to Kornberg's lab. He went to Kornberg's lab and learned how to make DNA polymerase and brought it back to Wisconsin himself. And now you see him actually using the DNA polymerase. Mouse pipetting, which is not allowed anymore, but at that time was, right? And he was working in the lab himself. So he was the work that led to establishment of the genetic code, he was personally involved in it. Not only the other people in the lab, but he was also working on it. Yeah. Yeah, so that's really. Now, the other thing I would say about Gobin is that he was an extremely humble person. All he wanted to do was learn. It doesn't matter whether he learns. He was a very established person now. It doesn't matter whether he goes to a lab, and of course you don't get to work with a professor. The professor is busy, even though he's a good friend of yours. He will say, okay, go and talk to so and so, and he will show you how to do it, or he will work together with you. So he was quite happy working with a lowly first-year graduate student, yeah? And then learning from him or her, and bringing it to his lab. And he did that not in Kornberg's lab, he did that in another lab to make RNA polymerase. And I know a student who showed him how to make RNA polymerase in another lab. And he was supposed to come here but couldn't, unfortunately, for this meeting we had here yesterday and the day before. So that was his motto was in science you learn from anybody, no matter who. And the idea is to learn. Okay, so this is now the genetic code. This is what really got him the Nobel Prize. And I think it was a great day. And this led to the Nobel Prize. And here you see the 3 Nobel Prize winners in that year in medicine and physiology. This is Robert Hawley. The 2 who won it for genetic code is Nirenberg and Govind. And this is the one who won it for establishing the sequencing of a molecule called a tRNA. But so anyway, the 3 of them shared the prize. And here we have the King of Sweden giving him the prize, a happy occasion. And now I show you the next slide. And this is a very interesting slide. So when you work for a scientific cause like that, establishing genetic code, it's a big problem. Right? They call it the Holy Grail. Everybody is after it. Yeah? And they want to be the first people to achieve it. And in science, when you really work, you put a lot of effort. You work hard, right? And then you also want to be the first to do it, right? Or people call it win, but it's not really a win. You just want to somehow feel that you've achieved something. And so that happens when 2 different groups are working for the same cause. There's intense competition. There's a lot of competition. And that can lead to some personal animosities and things like that, right? But in science, I think there's less such personal animosities than in other things like, And so this slide shows that, and as a good example of that. In 2009, we had a meeting similar to the one held here in Wisconsin, where he came from, where he was when he won the prize. And Marcel Nirenberg, this here, person here, and he are holding hands. Right. This was a really very touching moment. And the person you might recognize here is Asim Ansari. Asim was there. He had actually, he and I had put together the meeting, but mostly Asim had put together that meeting. And so this was a very happy occasion, but particularly touching was Nuremberg and Khorana holding hands. The two intense competitors for the genetic prize, genetic code work, holding hands, right? That's how science should work, right? And so he was, unfortunately, Marshall Nuremberg, pretty soon, he passed away about three months after this meeting. Now, this picture is an absolute classic, right? So I'll tell you a little story about this picture. This was a day of the Nobel Prize. Gobind was probably the last person to know about the Nobel Prize. He had, the reason for it is that he usually used to go, intense concentration, he doesn't want to get distracted. He was writing papers. And he wanted to write papers in peace, nobody bothering him. So he would book a room at a motel or a cottage somewhere where there's no telephones, no radio, no nothing, basically. And his wife would drive him there and then go pick him up whenever he wanted, prearranged, right? And so we all heard about the Nobel Prize. We all went to the lab very excited, but no Khorana to be seen there. And so he, eventually Esther, his wife, brought him back 10 o'clock, 10:30 or so. And then there was mayhem in the office when he arrived. But then pretty soon after, there was the Enzyme Institute was 3 big groups, Khorana's group and 2 other groups. The chiefs of the 2 groups decided to have an impromptu party. This is now the picture taken at that impromptu party. Right, for a party for Gobind and for members of his research group. And so what's he doing? He is now here writing on the board, talking to 2 faculty members. They're both from the biochemistry department. Not about how he won the genetic code. This is not about how he won the genetic code. This is about what he was doing right now in the lab, working towards synthesizing a gene. This is what it was about. And they're holding the champagne bottles. Everybody has got champagne glasses in their hands, right? But the science that they're talking about is not what was done maybe 2 years before, right? But it was what he was doing now. And this is the extent to which he's dedicated. When he's just absolutely single-minded almost. And that you have to be at times. So how was he going to put together a gene? All he knew was how to synthesize small oligonucleotides. But then gene is a big, big, big thing, right? And so what he did was to somehow bring them together and join them by using an enzyme that just joins DNA molecules. And this was the idea. So this is what he was trying to show here, trying to explain to these guys, right? How he would make these molecules and somehow join them like that or join them like that, right? And so this is it. And so this I show you again, right? This is one oligonucleotide here, another one here, another one here, and another one there. And if you mix the 4 together, they will come in the way that as shown. Only when there's no gap here, but there's a gap here, but a gap that can be sealed by the enzyme called DNA ligase. So you from starting with 4, you now make 1. And similarly, starting with, I don't remember, 1, 2, 3, 4, 5, 6. Starting with 5, you make another one. And now the 2 of them can be brought together because you have CTAA will go and combine with CTAA. And now that will join with that. And that was a so-called stepwise way of joining from starting with simple chemically made synthesized oligonucleotides, join them using an enzyme, and then you get the DNA of the size that you want to make. So this was it. And so pretty soon, it was published in 1970. And here's another thing I want to say about Gobind is he was an incredibly loyal person. Right? The reason I went a year before he did was he wanted to stay in Wisconsin to finish the work, publish it from there. Because the work was done there, he wanted Wisconsin to get the credit for it, that it was done there. So this was the paper. And then, so Wisconsin, in fact, this is such an important thing to have happened. This is really one of the most important things in genetics. The first thing, synthesis of a chemical synthesis of a gene. Wisconsin has a plaque commemorating that right outside the biochemistry department. Okay, so now in the paper that he wrote, I think I'll just point out to this one here. So you make this DNA now, right? You have small pieces, they made DNA, great. What are you going to do with it? You need a lot more of it than what you can just synthesize chemically. So now he was thinking already of how can I amplify this DNA? And so he wrote, at the end of one of the papers, he wrote this statement. The principles for extensive synthesis of duplex tRNA genes which emerge from the present work are the following. So this is his idea of how you would amplify it. The duplex DNA would be denatured to form single strands. You just, the two strands are together, you'll separate them, right, apart. The DNA duplex, this denaturation step would be carried out in the presence of a sufficiently large excess of two appropriate primers. And what he's saying is that you add then small bits of DNA that can bind to those long DNAs, right? And then you can now fill in the gaps. And then from one molecule of DNA, you make two molecules of DNA. And you repeat that again. From 2, you'll make 4. And from 4, you'll make 8. And then so you go exponentially go by twofold increases. And pretty soon, if that works, you make tons of DNA. Same singles, you make the same gene many, many times. Now, does that sound familiar to anybody in the audience? PCR, somebody said, right? Yeah, exactly, PCR. And so that was he had foreseen the use of PCR years before. Kary Mullis published his PCR work and got a Nobel Prize for it. And there are many, many people, I know many, many friends, not only friends, many, many scientists. I might be biased, but many, many scientists who feel that Gobind should have won that Nobel Prize too, shared in that Nobel Prize too. But he was there 15 years before Kary Mullis was. So this again, what I mean to say is that he saw things way before, long before anybody else did. Now, there's a lot more I can say about his work at MIT. Besides that, he went on to MIT. He went on to synthesize another gene, and then he went on to hook it onto a DNA that he can introduce into cells, and showed it was functional. And that was again. So I've already said that. Right? Now, so this is the principle of PCR, right? So now, of course. For a scientist of his stature, it's not surprising that he won the Nobel Prize. And he also got many awards and accolades and honorary degrees and all that stuff, which is great. But he was thankful for that. But to him, the important thing really was he wanted to be close to his lab. He was an extremely modest and a humble person. And he did not want to really just look for the limelight. He would much rather be left alone and be allowed to do things the way he wants to do. And so one of these awards actually is this shown here. He was also elected to the Papal Academy. The Pope in Rome has an academy, scientific academy, that there's a bunch of advisors advising the Pope appoint, and they bring in, I don't know how many, they probably have 10 new scientific advisors every year and things like that. And so this is one of those occasions where the Pope is basically inaugurating Gobind to that academy. Yeah. So, and that's very nice, but basically he was just someone who wanted to be left alone, do his things, and his motto was really same as that of Otto Loewi: we must be modest except in our aims. Right? So he was a very modest person. If you walked in here, no one would think that you had a Nobel Prize or anything like that. It's just a plain human being. But his aims were high. And whatever he tried to do in science, he aimed for things that seemed to be unreachable at the beginning. And yet he ended up by achieving those aims. And that was really what he was able to do. Now, what he really appreciated, as I said, is that to be with the students. To be with people who are interested in science. And to him, this was one of the most satisfying ends to his career, if you like. This was in 2009. Now, there is a program called the Khurana Internship Program. I suspect many of you don't know it, because there's a program that allows, at the beginning, about 15 students from India to go to the United States every year and spend a summer working in research labs. And this was initiated by Dr. Asim Ansari, who is not here today. He was here yesterday. And with the help of the Indian government, of course, who provided the funds, and also with the help of the University of Wisconsin, where Asim is now. University of Wisconsin provided funds, and that's how this whole thing started. And so that was the beginning. Now it has mushroomed into a big one. Now there are 45 to 50 students go every year. Under the Khurana internship, they also now started the Bose internship, another one, similar to that, based on the Khurana internship program. And now at that meeting then, this was the summer, it was held in the summer. So all the interns, Khurana interns were there, and he was really, really happy to meet them. This was the first time that he had met here. These are the Khurana interns who were there in that year. So this was a very happy occasion for him, and here he is really just talking and chatting with these students. Now, this is another happy occasion. Is so we go back to 1968 when he won the Nobel Prize. It was a day of parties, literally. And so after the party in the lab, Enzyme Institute, in the evening, Julius Adler, a really, really good friend of his, who also faculty member in the biochemistry department, gave another party for the Khurana lab and some other faculty in the biochemistry department. And it's traditional that if you celebrate with champagne, in the United States at least, and so they were drinking bottles of champagne. And Julius had saved one bottle that was autographed by Gobind, right? So that bottle from 1968. And so in 2009, when we had the meeting, Julius invited us to dinner. So we went home to his house for dinner, and he brought out that bottle, and he's just showing it to Govind just for memory's sake. And this is that's another example of simple things like that he would enjoy. And then he would enjoy this. Two of his students, women students, Kovan Kai was supposed to be here, but I got an email, Rajendra, saying why she couldn't come. Oops, sorry. Yeah, so Kovan couldn't come. She's in Shanghai right now. And this is Julius. Two students getting their degree that day. And of course, when his students are getting degrees, he wanted to also get dressed, so he got dressed. And now I show you something that you might be really interested in seeing. He would accept any challenge. Even challenges that you might kind of think that was not a good thing to do. And so, this was a lab outing. You go out, you have barbecue, you eat, and all that stuff. And someone challenged him to see whether he could put a bottle on his head and walk. And so this is him and a lab member. Yeah. So him and a lab member just doing that. There's a bottle here and a bottle here. Yeah. So that's and other things that he enjoyed is that now there is at the University of Wisconsin, outside the biochemistry auditorium, like our auditorium here. This is probably fancier than the biochemistry auditorium, but anyway, they were having a new biochemistry auditorium in 2009, and outside the biochemistry auditorium, they have named the auditorium the Khurana Auditorium, and this is what it says here. So he appreciated that kind of gesture. And he appreciated the fact that the town of British Columbia and the University of British Columbia in Vancouver would actually have a park named after him. So this is a park named after him. It's called, here it says the Khurana Park. Yeah, and a beautiful park. Yeah. And right next to that park is a park named after Mike Smith, because Mike Smith passed away also. Park after the person who was his student and who also won the Nobel Prize in chemistry. So that's the kind of thing that he enjoyed. Yeah. All right. So this is, I can tell you a lot more, but I think that you got a feel for it. I hope that this is something now. Now I should say something about myself a little bit, but I'm not going to finish it. That's for sure, because I don't think I can go through all the slides. But basically, as has been already mentioned, I come from Nepal. When I grew up, there was only one college in all of Nepal. Yeah, so you can imagine what the situation was like. One college in all of Nepal, and I went to that college. And I graduated, I got a BSc degree from there. And that college was affiliated with Patna University. So that's why my BSc degree is from Patna University. And then I was lucky enough to do well in my studies that I got a scholarship to go to Calcutta for my master's degree. And the scholarship was a scholarship that at that time, just soon after independence, Pakistan, India, Ceylon, Burma, all the countries became independent. So they formed a kind of a commonwealth. That became part of the British Commonwealth, right? And they had a meeting in Sri Lanka, what is Sri Lanka at that time, Ceylon, in Colombo. Colombo Plan. So that was the beginning of the Colombo Plan, And so somehow Nepal was never a part of the Commonwealth, but they thought the Nepal needed some help. And so they different countries offered help to Nepal. And so I came under a Colombo Plan scholarship to Calcutta to do my master's degree. And I had 2 very nice years in Calcutta. And I was fortunate to do well there. When I went back, I could get another fellowship, Colombo Plan fellowship, this time from England. So I got to England. I had no say, like Gobind, I had no say in which lab I went into. It's just that the British government would somehow find a place for me. That's all I knew. And I ended up in the lab of, okay, so this is my path in science, I should mention. Yeah, so from Kathmandu to Calcutta, Calcutta to Newcastle, where I did my PhD. Then I went to Madison, where Gobind was, to join him, and then to Cambridge, MIT. Yeah, so that's basically my path. It's a much shorter path than Govind's, but in any case similar to his, because we both spent a good bit of time in England. We both were started in chemistry. My PhD is in chemistry. We both were chemists who went into biology, and we had many other things in common. And that's really what brought us together somehow. I had the privilege of really knowing him so well. Okay, so and then this is the professor I work with, Sir James Baddeley, and a wonderful person. I think I learned a lot on research from this man. He also passed away just a few years ago, but. So what was my PhD thesis titled? My PhD thesis was titled coenzyme A. I was going to synthesize coenzyme A. And as I was working towards it, there's about 11 steps of a 14-step chemical synthesis. I think I was on the 11th step. There was a paper from Khurana's lab, synthesizing. They had completed the synthesis of coenzyme A. so the title of my thesis changed. a little bit. It still said coenzyme A. But so anyway. But then that was yet another connection between me and Gobind. That somehow coenzyme A brought us together. And the animosity that, any animosity that was completely vanished. And so now, let me just tell you a little bit about what I have spent all my life on. I'm not going to go into details at all now, because I don't think we have time. So, for our students, I just want to just bring it back to you. You probably know it all, but it never hurts to kind of recap on things. So we have this thing called central dogma they call it central dogma of molecular biology, or central dogma of biology, whatever you like to call it, right? So it says that basically you have the genetic information is present in the form of nucleotide sequences in DNA. That information is replicated in a process called replication, is copied to make more DNA. That's how the information is passed on to successive generations. And that information, if it to be used, for, let us say, to make a specific protein like hemoglobin or whatever, right? The information in DNA would be transcribed to RNA, messenger RNA, And the messenger RNA would be translated to protein. So we have the information DNA is in the form of nucleotides, ACGT. Information RNA is also in form of ACGU, which is U and T are basically the same. So it's basically the same things. Information in the form of nucleotides, and that information is translated to protein. Protein has not nucleotides, but amino acids, so it's called alanine, leucine, tyrosine, etc. 20 or 21 different amino acids. So that's basically what happens. And so I have worked all my life, literally, on this process of messenger RNA being translated to protein. I started on this work because when I went to Govind's lab, we talked as to what I would be doing, because the tRNAs play a very critical role in this process. And I'll tell you how tRNAs work in this process, but this translation of information in the form of nucleotide sequences to amino acid sequences of the protein is called translation, of course, and then it involves ribosomes, translation factors, and a whole new class of molecules called tRNA or transfer RNAs. How do transfer RNAs work? Transfer RNA work as follows. If we have a sequence in the messenger RNA called UGC, GUG, CGC, and UCG, and let us say GUG says, I want valine there, right? And that is done by bringing a tRNA which binds to that GUG sequence, C to G, A to U, and C to G. Again, the same Watson-Crick base pairing scheme, right? And that tRNA would now bring the amino acid valine to the ribosome, which is where proteins are made. And then when that has happened and this valine has reacted with the previous one, then the next tRNA to be brought would bind to GCA, and that will bring alanine. So in other words, this tRNA is acting what is called an adapter. It's acting as an adapter between the nucleotide sequences on the messenger RNA and the amino acid that messenger RNA signifies for that. So it's a codon. These things are called codons, and that sequence in the tRNA is called the anticodon. And what Govind showed was that GUG stood for valine, GCA stood for alanine, UCG stood for serine. Now that is the genetic code. Right? So that's really it. And so I was really enthralled by this molecule called tRNA. And so basically, tRNA help you to change the language of nucleic acids to language of proteins. So now, if you look at this, then if you were to change this, for this tRNA has to be called amino acid. It should be attached only to valine, not to any other 20 other amino acids. So the question is, how does that happen? How can an enzyme that attaches an amino acid here, right, know that it's to attach the amino acid valine only to this tRNA, not to all the rest of the 40 other tRNAs? So this means that there are proteins recognize RNA molecules with incredible amount of specificity. If it's not specific, you're gone. Nothing works, right? So that to me looked as if this was an incredibly interesting molecule to work with. And fortunately, that's exactly what Govind wanted me to do when I first went there. Govind wanted me to work on tRNAs because that's what he had been working on. He passed it on to me because so he could work on the genetic code himself. And so that's really what has got me to work on tRNAs. And I think, you know what, I'm not going to go into this because this is not, yeah. So over the years, then I have had the pleasure of working with many, many people. I had many people from India, actually. I have, one of them is in the audience here, Umesh Varsne. Umesh did things in my lab that kept me going for at least 10 years, if not more. You know, he made such discoveries that helped me so much, right? So I want to really. And then there was another person from Bangalore who trained for his PhD in Bangalore. He came to my lab, did phenomenally well. Another person came from Pune, came to my lab, did very well. All working on this small project. I was just going to talk to you about the project that Umesh and Ramesh and Sadanand and others will work on. Yeah. So the people who worked with me on a project. Okay, so these are all this. It's a shame, but I'm sorry, but Yeah, I'll tell you. I will go into this a little bit. Yeah, First, I'll tell you who these guys were who work in the lab, and then I'll go back to it, because at least our students should know at least that, the difference between basic research and applied research. I just want to say a few things. Okay, so these are the guys who work on that particular project. That is very thin slice of the work that happen in the lab. And Ramesh from Bangalore, Saranand from Pune National. Nripendranath Mandal from Calcutta, Barry Seong, Baeklin Seong from Korea, Shamping Lee from Taiwan, Deb Mangru from Canada, Mike Dyson from England, Christine Meyer from Austria, and Carolina Kerrer from Austria. So these are the people who work on that particular project. These are all actually postdocs. Right? Yeah, these are all postdocs. But they helped me greatly in this one big problem that we were working on. It took many years, but I think it was very satisfying. So now one thing I should add is the following. So there's a lot of talk these days about what should one be doing. Should one go into applied? Should all research be really applied? Or should you also be doing basic research? And I believe strongly that unless you do basic research, unless you have the basic information, you can't really do applied research. So basic research has a very, very important part to play. And now I'll give you two examples of where basic research can, I'll just give you just two examples so people can understand it. One basic research that was done, one thing that was done was this one here. At one time, what we had to, so the tRNAs bring an amino acid, and there's one class of tRNA called initiated tRNA, where that amino acid also undergoes another change called formylation. Yeah. So we have tRNA goes to, you add an amino acid, and then you add another chemical group to that amino acid. So you have three different forms of the tRNA. And it became very important for us to know what is the form of that tRNA in the cell at one time, because we were making many kinds of different, Umesh was making many kinds of different mutants and things like that. And what Umesh showed, this is the second of his big discoveries. The first one is a different one. And maybe I'll go to it a little bit. Is that Umesh showed that you can actually separate this is the one form of the tRNA, this is the other form of the tRNA that has an amino acid attached to it, and this is the third form of the tRNA that has a formyl group attached to that one. He showed that these three forms can be separated. And by doing so, then if we can make a mutant tRNA and we can look at the cell, and just comparing that to that, we can tell what form that tRNA is in the cell, what form that tRNA is in the cell. These are the different forms of mutants, right? So if we look at G72 mutant, for instance, that is in the form of just the amino acid added to it. With the wild type tRNA is all in the form of the formyl amino acid. So it says that this mutant is blocked in the step that takes you from there to there. That's basically what it does. Yeah. So this is the big thing that was for us. Now, basic research. Okay, it's good for your project, but what else is good for? Now, well, I'll tell you something. Now, this is just a few years ago, about 4 or 5 years ago. A fellow named Vamsi Mosa came to see me. Vamsi is a MD, PhD. He works at the Massachusetts General Hospital. He'd been collaborating with these doctors in Australia who had been working with patients, children patients, who had a mitochondrial disease, and they had no idea. All they knew is that there was some problem with mitochondrial protein synthesis. There's also protein synthesis in the proteins are made in the mitochondria. And he had no idea, but he had just some clue that maybe it is blocked in converting a tRNA from one form to another form, because they found that they saw a mutation in a protein that would do that. But they were not sure. So Vamsi comes to me and said, well, can you help us? And I talked to him and said, well, maybe we can. Let's give it a shot. It was not a guaranteed thing. It was a long shot, but I had a particularly good person in the lab. Same person I mentioned before, Carolina Kerrer from Austria. And she did an experiment that actually proved the point and said that indeed this particular disease that the children are suffering from is caused by that event. And so now that you know it, now you can think of what else can be done too. Right, so this was the paper that was published with Vamsi. And it says that, it's showed here that patient fibroblasts have dramatically reduced FMET tRNA MET levels and an abnormal formulation profile of mitochondrial translator COX-1. So this is the data. The data is that we can separate, as I mentioned, a plain tRNA from FMET tRNA, that is formalin methionine, from methionine tRNA. That's what we can do. Now, if you look at to see what form this tRNA is in normal cells, normal human cells, it's in the form of two forms. tRNA MET, that's just got tRNA with no amino acid or anything, and this has got formalin methionine. So in the wild type, when normal human beings, it's in the form of FMET, and that FMET is what works in protein synthesis, initiation of protein synthesis. And now, if you look at what's going on in the patient fibroblast, you see that if in the patient fibroblast, patient one, there's no FMET, right? See, and there's no MET. So the patient one is completely blocked in the conversion of MET to FMET. And that's exactly what we wanted to see. And this is proof that patient one and patient two are, they're blocked in the same step of events. And so this is a very important thing, yeah. So, and Rajendra is pointing it out to me with my time. So the other thing is really that, you have mutations in a gene that somehow introduce stop codons into a tRNA, into a messenger RNA. And if that happens, then you don't, stop codons into messenger RNA. And if that happens, you don't make any of that protein because the protein synthesis translation starts here, and then it comes to a stop pretty soon after. Right? So you don't make any of that protein. But if you now add a tRNA to it, that can read that stop codon, which is not ordinarily in the cell, but you can make one and make it. That can now be read as a normal sense codon, and you'll make a full-length protein. Now, there are many, many diseases where you have Duchenne muscular dystrophy, cystic fibrosis, where you have what's called a premature stop codon, and that's the disease is called by that stop codon, and not making any of that protein. Right? And you can now think of ideas where you can either introduce a stop codon and cure that, or not cure that, but at least the potential is there. Or if you can somehow persuade the ribosome with little small drugs and things like that to read through that stop signal as a genuine signal, then you can also correct that defect. So the point is that if you learn about it, if you do basic research and know what is involved in reading through a stop codon, or know what's involved in introducing suppressor tRNAs into cells and things like that, you can think of correcting defects in the gene. So I'll just give you two examples that involve tRNA. There are many, many more. I'm sorry I could not go through my work, but I think I really wanted to talk more about Govind than myself, and I hope that it's been worth it. Thank you. Sorry. I go down now, right? I go down now, right? Yeah, it's okay. Thank you. Yeah. Thank you.
Mike.
Oh, thank you.
We are indebted to Professor Bhandari for taking us to incredible scientific journey of Professor Hargobind Khurana and sharing his experiences with us. Indeed, this was a thought-provoking and energizing lecture. Thank you, sir. Now, I would like to invite Professor Arun Grover, Vice Chancellor, Punjab University, guest of honor, to share a few words with us.
So we had a mesmerizing lecture from Professor Raj Bhandari, Tom Raj Bhandari, Uttam Lal, Tom Raj Bhandari. So I do not want to take the flavor of his lecture from all of you. So I've been asked to be brief. So let me extend an invitation to Sir to come back to us very soon, maybe a year or two from now, okay? Just on the occasion of, let me state it, Punjab University is one of the 5 old universities of India, which were there at the beginning of 20th century and which were regulated by the Universities Act 1904, the Bombay, Calcutta, Madras, which were the universities established by the East India Company. Then the 4th one for the region of Punjab, the region of Maharaja Ranjit Singh's Punjab. So this was a university created by trifurcating the University of Calcutta. First bifurcating the Calcutta into Calcutta and Lahore, then trifurcating Calcutta, Allahabad and Lahore. So these were the 5 universities of India governed by the then central government. Okay, and then more universities came in, and Patna University was making, 4th university out of the Calcutta University. Actually, the Patna College, Government College at Patna was established 1863. Lahore College at Lahore came up later in 1864, but the people of Punjab were restless. They campaigned to have the university. People in Patna did not campaign, so they didn't get the university. The Lahore got the university. Allahabad anyway was given the university because of its importance at the central provinces. So the Patna University came up in 1917 and Modi ji was there for the centenary of Patna. So 2019 would be a centenary of the sorts for our region. And what is that centenary of the sorts? The British commenced the universities, but they did not give them any money to appoint teachers for the universities. They said, here are the universities, appoint teachers, but no money. Money was to come up from the profits of the examination fee from the students who wrote the college entrance exam and the students who wrote the college exams, intermediate, as you mentioned, okay, class 11, 12, then BSc, BA, etc., etc. They didn't give any money. So it was to come out of the profits of that the universities can generate. So universities were supposed to be financially self-sustaining, generate the money and run the universities. So when did the Punjab University start appointing teachers? Punjab University started appointing teachers only from the year 1919 onwards. So 2019 will be the centenary of having teachers on behalf of University of Punjab or all the universities of Punjab. So what did the Punjab University do when they appointed teachers on behalf of the university? They said university teachers would teach the chosen students drawn up from the colleges, okay, and the teachers and the students will learn together. And every student who passes through the university teachers would be given some exposure to extending the frontiers of knowledge, that is, do some bit of a research and if possible also write a paper before you graduate out of the university. So that is the start of honor school system of education on behalf of University of Punjab at Lahore. So this was the USP of University of Punjab at Lahore, and it continues to be the USP of the Punjab University campus at Chandigarh. The ISA Mohali, which is just diagonally across Nabi Mohali, is a reincarnation or a newer version of the honor school system that was commenced at Lahore. So every honor school student was expected to write a paper before you graduate out. That is how Hargobind Khurana wrote a paper working with his teacher before he passed out of the University of Lahore. So it's not only Khurana wrote a paper before you, another Nobel laureate who was a graduate and a contemporary of Khurana at Lahore, who passed out from the university almost the same year as Khurana, was Abdul Salam. Abdul Salam did mathematics with a great teacher, You perhaps know him, Sarvadaman Chawla. Sarvadaman Chawla, after the partition, went to Princeton first, Institute of Advanced Study, and then later on for all his life he worked, like Khurana worked maybe at Wisconsin and British Columbia, Wisconsin and MIT. He worked at University of Pennsylvania. He passed away a few years ago. So Khurana's, this Sarvadaman Chawla student was Salam, and he also wrote a paper before he write this thing. So this is the legacy of this. So 5 years ago, the students who are here, they are Har Gobind Khurana scholars. The Punjab government came out with a scheme that they will incentivize some students who are from the rural areas of Punjab, and they would be given scholarship so they can pursue their education first through the class 11, 12, and then so on and so forth. So in addition to this Khurana scholars, the young people for whom these lectures are held, the government of India also has a new scheme which is called an Inspire scheme. Inspire scheme is getting the top school students, get them together, have them, give them expository lectures, and these, these ISERs have been created to have further smaller subset of them and put them onto careers in sciences, careers in fundamental sciences, so that the science research has no national boundaries. So to give those people who are engaged in pushing the frontiers of science globally, young people to work with. So Inspire scheme is India's contribution to have young people to work with professors globally, so the frontiers of science continue to expand, so the benefits of science and technology can be shared by the entire human this thing. So in 2019, we will commemorate 100 years of having this kind of an education. We have Inspire camps couple of times in a year at Chandigarh. We had Roger Kornberg visit us last year, and address the Inspire students at Chandigarh. So I would like to extend an invitation to you to come here either next year or year after next when we are commemorating centenary of honor schools system. We are going to have a centenary symposium at Chandigarh on behalf of the Crick institutions and Punjab University, and maybe several more events connected with the commemoration of that centenary. So please do come back again, and we'll have an occasion to listen to you, your contributions to science, which are as noteworthy as those of that of your mentor. So let me conclude here. I know your flight is waiting and you have to leave. So thank you very much, and please do come back and be a part of us.
Thank you, sir, for sharing your views with us. I would like to invite Chief Guest of the Day, Dr. Roshan Shankaria, IAS, Principal Secretary, Science, Technology and Environment, Punjab, to kindly share a few words with us on this special occasion.
Good morning, everyone. We had the privilege of having talk from Professor Raj Bhandari, who was a very close associate of Har Gobind Khurana. I think it's a lucky day for all of us, and I extend a warm welcome on behalf of Government of Punjab to you, Professor Bhandari, to this, and we expect that you come in future as invited by Professor Grover. I was really pleased to go through the presentation made by you, and I think few things which are takeaway from this lecture are basically hard work, commitment, humility, and a focused kind of approach. People generally ask successful people, what is the secret of success? There is absolutely no secret whatsoever. It's only 99% hard work and just 1% inspiration. But of course that 1% inspiration also matters a lot. The kind of inspiration we got from today's lecture and going through the history of journey of Hargobind Khurana is, I think, really inspiring for all of us, and should stand us in good stead in near future to come. It's really amazing to know that the discovery made by Professor Hargobind Khurana, which was later on used for genetic re-engineering and all, has solved many problems of the humanity, especially the food problem. I have no hesitation in saying that Malthus, who gave a theory about 200 years back, he categorically said, he was a brilliant English scholar, and he categorically said that the population is growing exponentially, whereas the food production, that grows arithmetically. So beyond a point, the population will never rise. And about 200 years ago, the population of this planet was less than 1 billion. And today, through this genetic engineering, of which pioneer is Professor Hargobind Khurana in terms of discovering the genetic code, synthesizing artificial genes, and then giving the scope of genetic re-engineering for producing new genes. I think the food production has really gone up, and today we have surplus food on this planet. The only problem is of redistribution, and we have 7 billion population. So probably totally disproving Malthusian theory. But still I believe a lot need to be done. You see on this planet there are many people who are happy, and there are many people who are not so happy, or maybe unhappy. And I think this depends upon the person, you know. A person may become even of the highest level in the world, you can say, but still he may not be happy. So what I believe is probably there is something intrinsic to a person which makes a person happy or unhappy. Maybe I think it has to do something with the imprinting of DNA, the genetic code, which you need to decipher. And the research you are carrying on regarding this transfer RNA, stopping genes and all, probably that can help us. How is that, you know, maybe those who are happy, their genetic code can be studied. Maybe we should be in a position to isolate or identify some of the genes which make a person happy. And then through this DNA polymerase technology, genetic re-engineering, we can have those kind of happy genes and get implanted into the future generations. Or the other way around could be those who are unhappy, their genes, they can be isolated. And through use of your technology, transferred tRNA, and stopping those genes from replication, I think those unhappy generation can be stopped. So I think still there is a huge challenge. I am saying it lightly, but see, all ideas initially come like that only. You never know, after 1,000 years, we may be in a position to have happy genes and everybody is happy on this planet. But the question is, if everyone is happy, then what is the purpose of life here? You feel happy only when you go through a phase of unhappiness. So there has to be a good mix of happiness and unhappiness. So science has got tremendous potential. The idea of saying this is that, there is nothing fixed in this life. 100 years ago, we never thought of kind of lifestyle we have here. Even 20, 30 years back, we never thought of this kind of lifestyle which we have today through use of computers, internet technology. I remember when I was in academy, our director used to be father of the CEO of Microsoft these days, Satya Nadella. So he used to be our director. So he used to say that, when you go back in the field, by the time there will be a technology whereby simply by clicking a button, you will be able to send letters from here to anywhere in the world. And it was so, unbelievable for us that a letter, how can you send it, across the globe and just click of a button? But now that has happened, email and all, and then connectivity. plus use of computer even in the genetic re-engineering process, I think it is possible to have the sequence of all genes, and based on that, there are many commercial organizations which are producing those artificial genes. So, I mean, the research has further been facilitated through use of technology. So what I'm saying is there's absolutely no limit to the boundaries of human thinking, human mind, the ingenuity of the human being. And we have all young, budding generations. I think today's generation, they start from where we stop thinking, that is the level difference, we can say. And these generations will definitely make a huge difference to the humanity. And that is possible if we have these kind of interactions more often. And I think really that will be a step in the right direction. And we hope that, the scientific community that responds to, all kind of challenges which the world is facing. On behalf of Government of Punjab, I would just like to say that, this particular place where this NABI is situated and other institutions are situated, ICER, INST, or CAB, the government has allocated 400 acres of land, 400 acres. And today, even if the cost of 1 acre is 100 crore, this becomes around 40,000 crores. So that is the kind of investment the government has made for the future generations to carry on with the education, higher education, quality education, research in science, biotechnology. Situation is quite different. When Hargobind Khurana, he grew up in one of the village where there was only one school, he had to go from here and there. So here the facilities are available, and I invite all the students and the people of Punjab to make use of these facilities. These are wonderful institutions, and Indian mind is intelligent, but unfortunately today, whatever facilities we enjoy, whatever facilities, maybe electricity, maybe TV, maybe computers, or any other kind of modern advancement you think of, has not come from the Indian side. We are very good in copying, replicating DNAs. So time has come whereby the original thinking should come in, and our future generation, they should strive to, develop that original thinking and come out with original ideas through basic search, which is useful to the humanity. So without taking much of the time and without letting your flight go, Professor Raj Bhandari, I thank you and the organizers of this conference, and giving me also a chance to have an insight into the life of Hargobind Khurana, the real genius. Not he discovered gene, but the real genius because of that. Thank you.
Thank you, sir, for your inspirational words. I also request you, sir, to kindly felicitate